Hey, and yeah, hi everyone. I'm Jerry Zhang. I'm a postdoc at University of Chicago, and thank you for the organizers for giving me this opportunity to present our work on the comprehensive analysis of a large human gut Bacteroidales culture collection. So I guess this audience does not need more introduction on human gut microbiome, but as you can tell from the thesis, basically a microbial profiles of fecal samples of 21 healthy donors from New York and Chicago, and as you can tell, it contains many different families, genus, species. But those, as you can tell, color in cyan belongs to Bacteroidales order, and Bacteroidales is an abundant as well as a core order within the human gut microbiome. And the species within the Bacteroidales has been repeatedly associated with human health and diseases such as obesity, type 1 diabetes, cardiovascular disease, autoimmune diseases, etc. But also the Bacteroidales species encode interesting functional genes and properties such as carbohydrate active enzymes or cazymes, the type 6 secretion system, as well as production of a variety of secondary metabolites. What's more interesting, though, is that different strains within the same species of Bacteroidales would have contrasting phenotypes, such as butyrate resistance or resistance against type 6 secretion system through acquired interbacterial defense or AID. Moreover, a study has shown that Bacteroides fragilis, a species within the Bacteroidales, would undergo extensive evolution in a single person over the years. So our questions are, what is the genomic and metabolomic diversity of human gut Bacteroidales? Not as genus, not as species, but actually at strain level. And besides, what evolution would have the Bacteroidales undergone in a co-residence ecosystem that is in the same gut? The Dutch Swab Family Institute, or DFI, has established a symbiotic bacterial strain collection, or biobank. We have recruited healthy human donors and collected their fecal samples. And upon anaerobic isolation and whole genome sequencing, we have isolated 408 Bacteroidales isolates, with which we have performed comparative genomics, metabolomics, as well as gene transfer and strain evolution analysis. Before jumping on all 408 Bacteroidales isolates, which belong to 9 genera and 35 species, we have first performed a deduplication based on ANI, SNPs, and indels. This reduced to 127 unique Bacteroidales isolates. From this phylogenetic tree, you can already tell that we have isolated the most common Bacteroidales species, as well as the less characterized ones, such as Alice diabetes, Odoribacter, Butyricimonas, as well as many species within the Prevotalaceae family. Just by looking at the genome sizes of these isolates, you can already start to appreciate the huge interspecies and intraspecies diversity. For example, we have many isolates of Phocaecolobulgatus, and you can tell that their genome size differences can be as large as more than 1 million base pair. We then further perform gene clustering of all the genes within these genomes, and we have plotted the isolate on this UMAP based on the presence or absence of these gene clusters. Even though, say, isolates from the same species tend to cluster together, you can also see and also appreciate the huge intraspecies diversity, for example, again, among the isolates of Phocaecolobulgatus. This also applies to specific genes within these genomes, such as cazymes. So we have enumerated the numbers of cazymes within each genome, as well as calculate the diversity of their cazymes based on their profiles. In general, the generalist, which can degrade more types of glycans and have more higher numbers of cazymes, and they would also have higher diversity of cazymes, and specialists vice versa. But you can also see that for isolates of Pivogatus, again, even though they have similar numbers of cations, the diversity based on their casion profiles differ a lot. Again, we can plot this based on a UMAP, based on the presence or absence of each casion families within each genome. And you can also appreciate that while the generalist tends to cluster together based on their casion profiles, the specialist would have a continuum or spectrum of casion profiles, and presumably varying glycan utilization abilities. The Bacteroidales also encode type C secretion system for interbacterial competition. And human gut Bacteroidales actually have 3 genetic architectures, G1, 2, and 3. They are mutually exclusive, except when there's defect present in one of them. Indeed, we see several isolates in our collection that contain, both GA1 and GA2 in their genomes, but inevitably we also found defects within GA1 of each of these isolates. However, we also observe very peculiar isolates, DFI 5.35 of B fragilis, which contain all 3 types of GAs within its genome. Even though we, again, observe a potential defect within its GA1, we did not observe apparent defects in GA2 and GA3. And why they can coexist and how they impact the phenotypes of this isolate needs further investigation. We've, of course, have also performed DNA transfer and strain evolution analysis on these isolates. For example, we have observed intergenous DNA transfer through large conjugated plasmid or type C secretion integrated conjugated elements between Bacteroides and Foucault cola. But we have also observed an interfamily DNA transfer between Odoribacter and Foucault cola of phages and AID. We have also analyzed intra-donor strain evolution. For example, we have isolated 4 different strains of B theta from one donor, but they appear to be derived from a single ancestral strain. And they evolved into 4 distinct strains by acquiring different plasmids, mobile genetic elements, as well as phages. Besides genomic analysis, we have also performed quantitative as well as semi-quantitative metabolomics on 111 unique isolates. In this heat map, each row is 1 isolate and each column is 1 metabolite. And you can also appreciate from a bird's eye view that there are interspecies as well as interspecies diversity in production or consumption of these metabolites in vitro. One particular metabolite that we're interested in is phenylacetate, which is recently shown to be produced by certain gut bacteria through oxidoreductases, and its level is linked to the cardiovascular diseases in humans. Indeed, we also observe a similar phenomenon in that the numbers of FAD-dependent oxidoreductases in the genomes of B theta or B uniformis correlates with the production level of phenylacetate in vitro. So to summarize, we have performed an order-wide comparative analysis of human gut Bacteroidales isolates, including those that are less characterized. And then we have reviewed inter- and intra-species genomic and metabolomic diversity, as well as strain evolution. For the sake of time, we didn't show here, but we also observed that the metabolomic diversity deviates from the phylogenetic diversity within our human gut Bacteroidales collection. The intra-species metabolomic diversity that we observe here highlights the necessity to functionally characterize the human gut isolates at strain level. There are many more details in our studies, and you can see them in our preprint on bioRxiv through this QR code. But you can actually also request individual isolates from our collection in this link or through this QR code. I'd like to acknowledge all DFI members, but especially Cody, Michael, Eddie, as well as Eric Paymer and Laura Comstock for their significant contribution to this project. Thank you, and happy to take any questions later.